that is normally found in the cytosol. So think about a protein that normally functions in the cytosol.
Okay.
So we just talked about the proteasome, for example. That's a protein that resides in the cytosol. Those proteins would not have a signal sequence on it. And when proteins don't have a signal sequence, the message is that they stay in the cytosol. If a protein has an ER signal sequence, then the message is that gets transported into the ER. So that's kind of like standard. And so then the question is, are they necessary for localization? Meaning that when you look at an ER protein, is that the part of the protein that's required to get it transported into the ER? And so to test that, what you would do is simply remove the ER signal sequence before the protein was transcribed and translated. So you'd have to do that like at the DNA level. You would just remove those nucleotides or mutate them, and then ask where the ER protein ends up. Does it still go to the ER, or does it stay in the cytosol? And so when they did that experiment, so you remove the ER localization signal sequence, the ER protein is now found in the cytosol. It's no longer found in the ER. So that suggests that the ER signal sequence is necessary to transport or to give instructions for that protein to become localized to the ER.
Okay.
That's the necessary part. The sufficient part is if you take a cytosolic protein and then you remove you basically add on not an NLS, you add on an ER signal sequence. And if you do that, then that cytosolic protein will be found in the ER.
Okay.
But that's true for any protein. So if you had a protein that should be in the nucleus and you removed the nuclear localization signal sequence, that protein can no longer get into the nucleus. It will stay in the cytosol. So a mitochondrial protein, if you remove the mitochondrial signal sequence, it stays in the cytosol.
Okay.
So that is like transferable to all of the signal sequences that we talked about. Likewise, you can do like lots of crazy things. You can take a protein that should normally be found in the nucleus, put on a mitochondrial signal sequence, and then it will be found in the mitochondria. So you can do kind of like weird things and then ask what happens when you change the localization of these proteins. Like crazy biology experiments.
Thank you.
Other things here that this is actually the experiment that removed, in this case, an NLS, mutated the NLS, and then instead of the protein being found nuclear, which this is showing, it was found cytosolic. So it's pretty powerful, actually, just mutating or changing an NLS sequence. And then, of course, like
That's
this
like the
general pathway of like nuclear transport, which is confusing. But knowing kind of the general process. Obviously, you won't have to draw it because it's multiple choice. But like the idea that there is a nuclear protein with an NLS that has to bind to a receptor. And then once it binds to the receptor, that gets transported into the nucleus. But the problem is it's like attached to the receptor. And that's why you need RAN-GTP. So that RAN-GTP can bind to the receptor, alter the conformation, and then that nuclear protein then can be released. And then that then signals the receptor to exit out into the cytosol to be reused. But then you have to get off RAN-GTP, so you do that by RAN-GTP undergoing hydrolysis, which then boots off the RAM protein and it frees up the receptor. to bind to another nuclear cargo protein.
So for the nuclear pore, is that open at all times to allow that to get through, or does it need a signal itself, or does it need the, like, channels or whatever?
Yeah, it's a good question. The nuclear pore is like a big mystery. So, like, we know this is kind of what it looks like. You have to think about it like that's just like a cross section. So you would have like two together. And that's kind of a pretty good depiction of what the nuclear pore looks like. So it's not like it opens and closes like a channel.
Okay.
It kind of always looks like this. But it has this like meshwork of unstructured domains, so proteins that have unstructured domains. They're composed of phenylalanine and glycine free repeats, so it kind of forms this mesh that basically prevents things from just freely going in and out of the nucleus. But for things like that, to my knowledge, is not clear. I've never heard a good explanation for this. Something about the nuclear receptor binding to its cargo protein allows that to pass through that mesh. So there's something about this arrangement that makes it interact with the meshwork and allows it to kind of wiggle through. It sounds like there's a lot that's like not worked out there, right? Like there's probably a mechanism that we just don't know about.
Right.
But we do know it prevents things from just randomly going through the nuclear pore. So we know things can't just pass. It has to be bound to a receptor, a nuclear pore receptor, which is very different than a plasma membrane receptor. These are like receptors that are in the cytosol that can move from the cytosol into the pore.
Got it.
But it's not really clear how that's happening. So it's just one like just the right interactions, and it somehow it wiggles through. But yeah, big unknown. It's a good question.
Cool. Thank you.
So I'm on the next lecture now. I'm on the protein codings. Do you think we could go over the Sex61 experiment again? I just think I'm kind of confused on that, how they discovered
Yeah,
it.
this is definitely a difficult experiment. Yeah, and maybe now that we reviewed kind of like what these signal sequences do, maybe it will make a little bit more sense, because that's really key to understanding this. So like, I guess the big picture question is, people wanted to understand or wanted to identify proteins that were involved in the secretory pathway. And so one of the first experiments that were done to identify these proteins was essentially this one that I'm going to describe to you. And they knew that the beginning of the secretory pathway lied in getting proteins into the ER, right? So every protein that is in the secretory pathway begins its life by getting transported into the ER. So they wanted to ultimately identify what proteins are involved in that process. And so they needed a way to determine if a protein was required to get into the ER. And so they wanted to identify genes involved in that ER translocation. So they thought of this like super cool, interesting assay, very smart, where they took yeast, so this was all done in yeast. And they had already known of a molecule called histidinol that was converted into histidine, which is an amino acid, because of a cytosolic enzyme. So there's normally, before anything happens to the yeast has an enzyme, and it converts histidinol to histidine. And the yeast use the histidine, and they're happy, and they can live. So they thought, okay, what if we totally like re-engineer this yeast cell such that we put an ER localization signal sequence onto this enzyme that's normally cytosolic, such that it should get shuttled into the ER, because we know it's sufficient, right? We know if we put an ER signal sequence onto this cytosolic protein, it will go into the ER,
Right.
because they had known that from previous studies. So the problem is that if this enzyme goes into the ER, it's no longer in the cytoplasm. Thus, histidinol cannot be converted into histidine. So now the cell will die unless the scientists give the cell histidine. Like if they put histidine in the media, then the cell will be okay because it has histidine externally, right? It can enter into the cell. But if they don't supplement with histidine, then those cells are going to die because this enzyme is now in the ER. Does that kind of make sense?
Yes.
Okay, so that's your assay. So then what they actually then would do, this is like tricky, is
Right.
so this is the cell they worked with. This is like their engineered cell. Then what they did is the forward mutagenesis screen on this cell, the engineered cell, where they, again, you just mutate genes, don't know which one you're hitting. And they took the yeast and they didn't grow it on histidine. And they reasoned that any yeast cells that could actually survive without histidine must be because they got a mutation that actually disrupted ER localization such that now this enzyme could be found in the cytosol and not exclusively the ER. So that if you kind of like disrupt a protein that's required to get proteins into the ER, then now this green enzyme can actually, sometimes at least, be found in the cytosol. If it's found in the cytosol, then it can convert histidine to histidine, and the cell lives.
Okay.
Does that make sense?
Yeah, I mean, it is just a confusing process, but it does help going over it again because it was a while ago. So.
Yeah. It's very, it's confusing. It's the cells that live that have the mutation that you want. And they live because the logic, so like genetics is all about logic.
Mhm.
The logic is that the only way they could live is if this green enzyme was not getting in the ER. And the only way it wouldn't get in the ER is if one of these proteins that is required to get that protein in the ER, one of those channels or one of the receptors or a chaperone or something, one of those proteins have to be disrupted that prevents it from getting in the ER. So therefore it stays in the cytoplasm. And so they identified a whole slew of these genes. One of those was SEC. So SEC stands for secretory. 61 was like the 61st gene they found in the screen. And it turns out that SEC61 is a gene that forms this translocator. So remember that the way that proteins are normally targeted to the ER here is that the protein is being translated, exposes the ER signal sequence. The SRP molecule binds to the ER signal sequence, and it gets transported, binds to the receptor, gets transported to the translocator. That's the channel that the protein then feeds through as it's getting translocated. As it's getting translated. So this would be like equivalent to the rough ER. But it has to bind to this translocator, and that's how it gets kind of fed through into the ER. And so SEC61 is a protein that's in that translocator. So that's actually the protein that they identify. And that's how we know what these proteins are. So we actually know the exact gene that's involved in that. And of course, we have the same homolog because these are really conserved processes. So this is like an example of like the SEC61 gene. And it was found in yeast, but humans have the same homolog and it has the same function. So yeast are really amazing systems because most genes of the secretory pathway were discovered in yeast. And of course, like I was saying in class yesterday, like nearly all genes of cell cycle, all those oncogenes and tumor suppressors, a lot of them were identified in yeast as well. So a pretty cool model system.
Thank you.
Yeah.